Seeds play an important role in the reproduction of pine, and we know that they're released from large woody cones. However, when these cones first appear in the springtime, they are small and fleshy. At this stage, they contain ovules and are called ovulate cones. The intact ovulate cone is seen to consist of numerous spirally arranged scales called ovuliferous scales. A closer look at a single spiral reveals that the cone is a compound structure. For each ovuliferous scale has a small bract fused to its lower surface. The upper surface of an ovuliferous scale contains two ovules. Each ovule has an outer protective layer or integument. An opening in the integument, the micropyle, consists of a canal that terminates in two micropylar arms, each covered with sticky secretion. Beneath the integument there is a fleshy megasporangium containing a diploid megasporocyte cell. When the ovulate cone is ready to receive pollen, the ovuliferous scales separate from one another, exposing the ovules within. Carried by air currents, some pollen grains are forced between the ovuliferous scales. Once inside the cone, the pollen grains fall down towards the ovules. Some of the pollen grains come into contact with the micropylar arms and adhere to their sticky surface. The megasporangium secretes a fluid which fills the micropyle. This is the pollination drop. Pollen grains on the surface of the micropylar arms are transferred to the pollination drop. The pollination drop soon recedes, leaving all pollen grains located on the surface of the megasporangium. At this time, the micropylar arms wither and the micropylar canal closes, effectively sealing the pollen grains inside the ovule. The scales of the cone grow together, sealing the ovules inside a tough, protective barrier. Once a pollen grain is on the surface of the megasporangium, the inner wall between the two wings begins to extend outward. The tube cell now forms a pollen tube, which grows at its tip and penetrates the sporangium tissue. The generative cell inside the pollen grain soon divides, forming a sterile cell and a spermatogenous cell. Both of these then move into the growing pollen tube. Inside the megasporangium, the single sporocyte cell divides by meiosis to produce 4 haploid megaspores. Only one megaspore cell is functional, and the others degenerate. As the megaspore enlarges, the nucleus inside undergoes repeated divisions, creating a multinucleate tissue. This is the young female gametophyte. Female tissue usually remains in this state over the first winter. At the start of the second growing season, whorls form around the nuclei of the female gametophyte. Several archegonia form at the micropylar end of what is now the mature female gametophyte. Archegonia are recessed below the surface of the female tissue. Each archegonium has a short neck. consisting of 1 to 2 tiers of neck cells. The large basal portion is the venter and contains a single egg cell. As the female gametophyte matures, pollen tubes continue their growth through the megasporangium tissue. When the pollen tube approaches the female gametophyte, the spermatogenous cell divides to produce 2 sperm cells. The pollen tube proceeds to grow towards an archegonium. Reaching the archegonium, the tip of the pollen tube forces itself between the neck cells and then ruptures, releasing the tube nucleus, 2 sperm cells, and the sterile cell. 1 sperm nucleus fuses with the egg nucleus to create a diploid zygote. If a sufficient number of pollen grains are present, pollen tubes deliver sperm to each of the archegonia within a single female gametophyte. The result is the formation of multiple zygotes within the same female gametophyte. At the time of fertilization, the ovule consists of several archegonia inside a female gametophyte, which sits inside a megasporangium. The male gametophyte has grown through the megasporangium to reach the archegonium. This is all enclosed within an outer integument. The following events take place in each archegonium that is fertilized. The diploid zygote nucleus undergoes 2 mitotic divisions, and the 4 nuclei migrate to the base of the archegonium. Successive divisions result in a 16-celled embryo. The tier of 4 cells at the base of the archegonium are embryo initials. The 4 cells of the adjacent tier are suspensor initials. The suspensor initials elongate, first pushing the embryo initials up against the wall of the archegonium, and then out into the female gametophyte, where breakdown of the adjacent tissue creates a corrosion cavity around them. The 4 initial cells separate, and each begins to form an embryo and additional suspensor cells, which push them further into the female gametophyte. Intense competition between the 4 young embryos for nutrients and space results in only 1 embryo surviving. Embryo systems and adjacent archegonia proceed through the same stages of growth and competition. Soon, however, 1 young embryo in the female gametophyte dominates and proceeds to develop further while the remainder abort. This surviving embryo enlarges and develops a shoot apical meristem, surrounded by a circle of seed leaves called cotyledons. Below the cotyledons, the embryo forms an elongated cylinder of cells that represents the stem-to-root axis. The tip of the root contains a root meristem, which in turn is covered by a protective root cap. During embryo development, nutrients accumulate in the female gametophyte, where they will serve as a source of nourishment at the time of germination. The final stages of seed maturation are accompanied by the removal of water from the living tissues, which then enter into a state of dormancy. The integument also undergoes changes, transforming into a tough, protective seed coat. Both the seed and surface tissue of the cone scale detach as a single unit. The mature winged seed is now ready to be released. When a seed is released from the cone and absorbs water, it is ready to germinate. Nutrients stored within the female gametophyte are now absorbed by the growing embryo via the cotyledons. Growth of the root results in its emergence and early functioning as an absorbing and anchorage organ. Soon, the stem axis below the cotyledons, known as the hypocotyl, begins to elongate. Continued hypocotyl elongation lifts the cotyledons out of the soil. When the seed coat is shed, the cotyledons expand and become green and photosynthetic. The short stem above the cotyledons, the epicotyl, begins to grow and to form leaves. And the seedling then proceeds to develop into another cone-bearing tree.
In conifers, the same diploid sporophyte plant has both male pollen-bearing cones called microstrobili and female seed-bearing cones called megastrobili. A female cone is a modified stem bearing a tight cluster of scales, which are reduced branches specialized for reproduction. A male cone is a cluster of scales that are modified leaves inserted on an axis. Female gametophytes are produced in the megastrobili, whereas male gametophytes come from microstrobili. Megastrobili are much larger than microstrobili. The megasporangium, in which the female gametophyte will form, is enclosed in a protective layer of sporophytic tissue, the integument, that will eventually develop into the seed coat. The integument, the megasporangium inside it, and the tissue attaching it to the maternal sporophyte constitute the ovule. The megasporocyte within the megasporangium undergoes meiosis to produce 4 haploid megaspores, only one of which is functional. The non-functional megaspores degenerate, while the functional megaspore divides mitotically, and the resulting cells divide again to produce a multicellular female gametophyte. Male gametophytes develop in a similar manner. The scales of the microstrobilus contain microspore mother cells. Meiosis gives rise to haploid microspores, which in turn mature into pollen grains, the male gametophytes. The production of male gametophytes in the form of pollen grains frees the plant completely from its dependence on liquid water for fertilization. Instead of water, wind assists conifer pollen grains in their travel from the microstrobilus to the female gametophyte inside the megastrobilus. The tiny pollen grain enters through a small opening in the integument at the tip of the ovule, the micropyle. The pollen grain germinates, forming a pollen tube. When the pollen tube reaches the female gametophyte, it releases 2 sperm, one of which degenerates after the other unites with an egg. Following fertilization, the sporophyte embryo begins to develop within the ovule, while the integument, not shown, matures into the seed coat that protects the embryo. The conifer seed contains tissues from 3 generations: the diploid embryo, the haploid gametophytic tissue, which supplies nutrients for the developing embryo, and the seed coat, which develops from tissues of the sporophyte parent, the integument. The seed eventually falls to the ground and, under suitable environmental conditions, germinates to form a new pine tree.